everyone, welcome back to Windows 8 Studies. I hope you all are doing well. So we were discussing about cell biology, in which yesterday we talked about transport of the molecules across the plasma membrane, and yesterday we specifically covered the passive transport, the transport of the molecules where energy is not required. The molecules just simply they move under the either hydrostatic pressure or according to the concentration gradient. Today we will be discussing about active transport, where the substances they move against their electrochemical gradient. So for that they require energy input, which is often harnessed in the form of ATP or from the pre-existing ion gradients. Okay, so today we will be discussing about active transport in detail. So we will be basically focusing on the types of active transport, their molecular mechanisms, and certain physiological examples of the active transport. So depending on like which type of energy is used. So we already know for the nature of molecules is that they want their potential to be equal at every point of time, and at every point of space given. For example, let's say that you pour water in a plate, so over the time it will flow in the whole plate, occupying the whole space equally. So the potential, so let's say that this is a container, okay, and you are pouring water from here, so water will accumulate here, but eventually with time it will flow and it will level itself accordingly. The more volume you put, but it is going to level itself accordingly. So this is the nature of molecules. It wants its potential to be equal at each point, right? So in the same way, the molecules in the cell, they also want their potentials, their concentration gradients, to be equal. Like there shouldn't be any concentration gradient. So this is the basic nature of the molecules. So let's say that this is a plasma membrane. So let's say this is a plasma membrane, and there would be some certain solutes dissolved in the solvent. So this is the extracellular matrix and this is the cytosol. So there would be solutes in the extracellular matrix, and we are assuming that this plasma membrane is permeable or semi-permeable, or let's say that it is permeable to this particular solute, which is in this case sodium ions. So if this membrane is permeable to sodium ions, by the basic nature of the molecules, sodium ions would want their potential, their concentrations to be equal on the both sides of the membrane. That is the basic nature of the molecules, right? But for maintaining the metabolism of the cells, for carrying out the activities, for example, we learned in the neurobiology classes that neurons, the cells, the nerve cells, they are the excitable cells due to the unequal distribution of ions on the either side of the membrane, and that what makes the cells excitable. So we discussed about this in detail in neurobiology lectures, right? So there we learned that for maintaining the basic cell functioning of the nerve cell, the excitability of nerve cells, you need to maintain the difference in the concentrations across the membrane, difference of the concentrations of sodium ions, chloride ions, and potassium ions. So any type of ions, the unequal distribution of them is very important for the basic cell functioning. And to maintain this unequal distribution, what you need is energy, because that is against the basic nature of molecules, right? So whatever is against the basic nature, you are going to need energy for that reaction. So whenever you burn anything, you don't need any energy, but whenever you have to reverse the reaction, you would want the energy. That is why for maintaining this concentration gradient across the plasma membrane, the energy is required, and that energy is provided either in form of ATP or already set ion gradients. So whether the energy that is used for bringing out this change to do this active transport, depending on which type of energy is used, there are two main categories of active transport. Okay, the first one we call it as primary active transport, and the second one we call it as the secondary active transport. Okay, so as I said previously that the energy could be harnessed in the form of ATP. So if the ATP is hydrolyzed And due to the hydrolysis of ATP, whatever energy is released, that energy is used to unequalize the distribution of ions or to set an ion gradient. That kind of active transport is known as primary active transport. Okay, so depending on the conformations of the transporters that are going to carry out the primary active transport, there are different type of ATPases, because they are the enzymes that are going to break ATP, and that is why we call them ATPases. So depending upon how do they do the ATP hydrolysis, there are different types of ATPases that are going to carry out the primary active transport. So those are P type ATPases, V type ATPases, or we also call them pump, because they are going to use energy to pump certain molecules. That is why we also refer them as pumps. And the third type is F type ATPases, right? And one more is ABC transporters. Okay, so we will be learning about all of them in detail. So let's start with the P type ATPases. Okay, so in the P-type ATPases, the letter P, it refers to the formation of the phosphorylation of the protein, the pump. So whenever there is a transport cycle, whenever the solute or the ions, they bind to those type of ATPase, and they want to, they want to cross the plasma membrane. Whenever this binding happens, this particular type of ATPase, this particular type of protein, that undergoes phosphorylation, and that is why the P stands for the phosphorylation in P-type ATPases. So this is a unique characteristic that belongs to the P-type ATPases. So let's discuss about P-type ATPases in detail. So let me just write it separately here. P-type ATPases. Okay, so the P stands for phosphorylation. Okay, so whenever there is a transport cycle, so whenever a solid binds to this ATPases, so during this transport cycle, the formation of phosphorylated intermediate is observed, and this formation of phosphorylated intermediate, due to this, those ATPases, they are referred as P-type ATPases. So those P-type ATPases, they have two subunits, alpha subunit and beta subunit. The alpha subunit, it usually contains ATP binding domain. It has a phosphorylation site, as well as it is usually the transmembrane domain. So it is going to hold the whole protein structure in the in the plasma membrane. The beta subunit, it could be there or it could not be there, so it is not mandatory to be in the conformation of the B type ATPases. So if the beta subunit is there in the protein, it usually is for stability. So it provides stability to the whole protein. It does the regulatory job. So it is also involved in regulation, and it is also involved in the proper folding. So it ensures that the protein folds properly, and it inserts itself in the plasma membrane properly. Okay, so beta subunit, it is optional. It may or may not be there. Okay, so though this P-type ATPases, they have two important configurations. They are referred as E1 configuration and E2 configuration, or let's call them conformations. So those two conformations, they have different properties. So for example, E1 conformation has higher affinity to the cytoplasmic ions, while E2 conformation has low affinity to the cytoplasmic ions. So the cycle, the transport cycles look somewhat like this. So let's say that this is a this is a plasma membrane, and this is the p-type ATPases. Okay, this is a p-type ATPase. Then what happens that a certain solid comes and binds to it. So let's say that this is a sodium ion and it is going to bind to it. So this is going, so whenever the ion binds to the p-type ATPase, that time the p-type ATPase will be in the even configuration, in even conformation, because even conformation has higher affinity, right? So this, in the even conformation, due to higher affinity, the ions come and bind to the ATPase. And once they bind to ATPase, so now they are going, so it is an ATPase, so it is going to hydrolyze energy. So it hydrolyzes ATP, okay, and due to hydrolysis of ATP, the phosphate is added on the pump. Okay, the phosphate is added on the protein, and the energy which is released, it brings about the conformational change from E1 to E2. Okay, so now the conformation of the ATPase is changed. E2 is a low affinity, E2 is a low affinity configuration conformation. So now the ion is released inside the cytoplasm or outside the cell. So whatever, so the sites are changed. So ion will be released to the other side of the membrane, because E2, this conformation has low affinity for the ion. So this is a whole. So once the ion is released, the conformation, the dephosphorylation happens, and after dephosphorylation, the conformation again changes to even. So when the phosphorylation occurs, the even state is changed to E2 state, and upon dephosphorylation, E2 state is again converted back to even state. So this is a whole reversible process. So this forms a cycle, a very nice cycle. So what happens is that even state, where which is a high affinity state, okay, due to phosphorylation, it changes to E2 state. Okay, and E2 state, which is a low affinity state. Okay, once the protein gets dephosphorylated, it changes from E2 to E1. So due to dephosphorylation, the conformation changes from E2 to E1. E1, which is again high affinity state. So this is how the cycle goes on, and this cycle is also referred as Albert's cycle. So this is how the transport cycle occurs in the p-type ATPases. So the very famous example of p-type ATPase is the one which we have discussed in neurobiology, the sodium potassium ATPases. Okay, so what does sodium potassium ATPase do? We have already discussed it. Let me just summarize it here for you. So sodium potassium ATPase pump, it does the efflux of three sodium ions. So let's say that this is extracellular matrix and this is cytoplasm. It does the efflux of three sodium ions and it takes in two potassium ions. So this maintains the concentration gradient across the plasma membrane. So what happens is that in nerve cells, okay, in nerve cells, there is an unequal distribution of ions. So there is which gives a total negative charge towards the inside of the plasma membrane and a positive charge to the outside of the plasma membrane. Why? Because the ions, they are unequally distributed. So all of the anions or the negatively charged amino acid residue, they are on the inside of the cell, and while on the extracellular part of the cell, there is sodium ion concentration higher and chloride ion concentration higher. So this gives an overall negative charge towards the inside of the membrane, and maintaining this unequal charge distribution, maintaining this charge separation is very crucial. It is very important, so that the signaling goes on in the nerve cells, and this it creates resting membrane potential, and this resting membrane potential is maintained by the charge separation, by the unequal distribution of ions, and this unequal distribution of ions is maintained by this p-type ATPase. this sodium potassium pump or sodium potassium ATPase. So in this sodium potassium ATPases, the amino acid residue which gets phosphorylated is aspartic acid, and this is a key question. This is very important one, because usually phosphorylation in the biological systems, it is seen on serine, threonine, tyrosine, right? We have learned about this in post-translational modifications in our epigenetic series. But in this case, the phosphorylation is done on the aspartic acid, on the aspartate residue, in the p-type ATPases. So this is very important point, that the sodium potassium pumps in this type of pump, the phosphate group is added on the aspartate residue. So this is one example. The other example of the same p-type ATPase, which you might know, is the calcium ATPase, okay, which is also referred as CSIR, because this is located on the membrane of the sarcoplasmic reticulum, means the endoplasmic reticulum of the muscle cells. They are referred as sarcoplasmic reticulum. So this is present on the membrane of sarcoplasmic reticulum. Okay, so this is present there. So what happens is that during the relaxation, so once the muscles are contracted, we already know that the calcium ions, they are crucial for the muscle contraction. They are important for the signaling so that the contraction happens in the muscles. So during that, all of the calcium which is stored in the sarcoplasmic reticulum, it is released inside the cytoplasm, and then muscle contracts. So we already know this, we have already discussed this in the neuromuscular junction video that we were learning about in the neurobiology. So we already know this. So but we have to reset it, like we have to, take the calcium back into the sarcoplasmic reticulum, and to do this, the calcium ATPase, this pump helps. So this helps in muscle relaxation. So once the muscles are contracted, now you want them to relax. So you need to take all of the calcium which was in the cytoplasm back into the sarcoplasmic reticulum, and that is done by the calcium ATPase. So this moves calcium ions back into sarcoplasmic reticulum lumen. Okay, from cytosol, obviously. Okay, so this is the job of the calcium ATPase. There's one more ATPase known as proton potassium ATPase, which is usually seen in gastric parietal cells, but we won't be discussing that in detail. So these are the different physiological examples of the P-type ATPases. So now the next one that we listed down here was the V-type ATPases. So all of them, all of those types, they are going to use ATP, but they differ in their conformation, or they differ in their in their working mechanisms, and that is why we have categorized them in different categories. So the V type ATPases, they are also known as vacuolar type. Okay, so let's just learn about V type ATPases. So V-type ATPases, they are also known, the V here, it stands for vacuolar type ATPases, because they are usually, they are usually present on the membrane of the, on the plasma membrane of the, sorry, plasma, not plasma membrane, on the membrane of the organelles. So they are usually present on the, vacuole type things. So vacuoles, what are vacuoles? They are basically the membrane structures that are found inside the cell, and they separate the compartment. So let's say that this is a cell, and this is the plasma membrane of cell. So it is compartmentalizing a portion, right? So it is separating the extracellular matrix from the cytosol. So again, in the cell, if a membrane does the compartmentalization, that type of thing is known as vacuoles, right? So the lumen part, it is separated from the cytosol part. So this is how the membrane does the compartmentalization. So on the membrane, on such membranes, there are certain ATPases that are situated, and they are referred as V type ATPases. This even though the V means vacuolar here, this they does not preferably are associated on the vacuoles. This simply means that they are present on the membranes that are found inside the cell, which are going to compartmentalize the cytosol and the lumen separately. Okay, I hope this is clear to you all, because this is very important, because they creates two separate environments inside the cell. So cytosol will be a different environment, and the lumen part will be the different environment. So for take example, take lysosomes. Okay, so in the lysosomes, you need acidic environment, right? So for maintaining this acid environment, the ATPases that are present on the lysosomal membrane, they are the V-type ATPases. Okay, so those they are present on the lysosomal membrane, endosomes, on the synaptic vesicles, because in the synaptic vesicles again you need to take neurotransmitter in, and that is how the V-type ATPases, they also help in neurotransmitter uptake. Okay, so this is how it has multiple functions. So the V-type ATPases, they have different conformation. So they have two domains, the V1 domain and the V0 domain. Okay, just like the P-type ATPases, which have alpha and beta subunit, the V-type ATPases have V1 and V0 domain. The V1 being the peripheral domain, and the V0 domain is the membrane embedded domain. So this is a transmembrane domain. Okay, so the V1 domain, it is usually the ATP hydrolyzing domain. So it has subunits which are going to do the ATP hydrolysis, while the V0 domain is the one which is going to form the proton channel. So the one thing that I want to mention is that V-type ATPases, they are always going to pump protons. Okay, so they are also known as proton pumps. So V-type ATPases, they usually have the role in acidifying the lumen. They usually uh, play a role in maintaining the proton gradient. So they are also, that is why they are also referred as proton pumps. Okay, so they just they just acidify the intracellular compartments and the extracellular environment. So wherever they are present, they are going to maintain the proton gradient, and that the maintenance of proton gradients help in the uptake of neurotransmitter, that help in maintaining the acidic environment in the lysosomes. So as we discussed about this neurotransmitter uptake, we have already been discussing this in neurobiology, but the one more example is the lysosomal acidification. So this is how the V-type ATPases, they are very important in the cellular functioning. Okay, so these are the details about V-type ATPases. The other type of ATPases, they are known as F-type ATPases, which are also known as ATP synthetases. So let's just learn about F-type ATPases a bit. So they are also known as ATP synthesis. So this is a bit different category, okay, because they function in reverse. Okay, so what happens is that they use the proton gradient to synthesize ATP. So basically they are also pump which are situated on the mitochondrial inner membrane. I'm sure that you must have learnt about this in your high school level. So even though we still call them ATPases, even though it does not hydrolyze ATP, but they are related to the active transport. They are not literally related to ATP transport, so the active transport, but they are mechanistically related to the active transport. So whatever mechanism they follow, which it is almost similar, and that is why we categorize this under the active transport. But I just want to point this out, that they are not the one which are going to use the ATP hydrolysis. They are actually synthesizing ATP by using proton gradient. So the proton gradient which was set by the proton pumps. So proton pumps are setting up the proton gradient. So now this same proton gradient could be used so that the ATP could be synthesized. So even though we call them F-type ATPases, but they are the ones which are going to synthesize ATP, and that's why the other name ATP synthase. Okay, so please remember that, and they are present on the mitochondrial inner membrane. So they are present on the mitochondrial inner membrane. Okay, I'm sure you have learned about it. This we just categorize them in the active transport because they are mechanistically related to that. Okay, and the fourth one, which is very important and which is also a type of active transport, is the ABC transporter. ABC transporter, that stand for the ATP binding cassette transporter. ATP binding cassette transporter. Okay, so this is very different because it has a lot of very different conformation from the P-type or V-type ATPase. So this is a large family of transporter which again uses ATP hydrolysis to drive unidirectional transport. So whatever direction of the transport was there, it is usually decided, it is predetermined, and they are going to again drive the transport against the electrochemical gradient. So they have two transmembrane domains. They have two transmembrane domains. Okay. And they also have two nucleotide binding domains. Two nucleotide binding domains. Okay, the transmembrane domains, they usually define the substrate specificity. So like to which substrate is the AABC transport is going to bind, that is usually defined by the transmembrane domains. While the nucleotide binding domains, as they are binding to nucleotide, means they are going to hydrolyze the ATP. Okay. So these are the main domains that are present in the ABC transporter. So what happens is that the substrate, whenever it binds to the transmembrane domains, okay, whenever it binds to transmembrane domain, that promotes the ATP binding to the nucleotide binding domain. And once the ATP binds to one nucleotide binding domain, the now nucleotide binding domain will dimerize. And after the dimerization, conformational change is seen in the outward facing state of transmembrane domain. So what happens is that the transmembrane domain, the inward face of transmembrane domain, let's say this is the inward face of transmembrane domain to which the substrate will attach. So this will promote the ATP binding to nuclear binding domains, and the nuclear binding domain will dimerize, and that is going to carry out the conformational change to the the outward facing state of transmembrane domain, and now the substrate will be released to the other side of the membrane. So this is the this model is also referred as alternating access model, but we won't be discussing about this in detail in this lecture. We'll discuss about this ABC transporter separately in some other video lecture. So what I wanted to convey is that the functioning of ABC transport is quite different from the P-type ATPases or V-type ATPases because it has a lot of regulation in it. It has clear-cut domains, and the similarity is that both all of them, they are using ATP. So ABC transporter, the very famous example of ABC transporters are the MDR protein, MDR1 protein, which is also P glycoprotein. So this is usually found on the bacterial membrane. So what does it do? It actually pumps out. So from the let's say that the bacterial cell has taken in the antibiotics, but the bacterial cell also has this MDR1 protein. Okay, if the bacterial cell membrane has this MDR1 protein, so now the antibiotic will be pumped out of the cell, and so if the antibiotic is being pumped out of the cell, that antibiotic can't work on its target. So some certain antibiotics, they target the DNA of bacteria, or if the genetic material is RNA, so whatever, or sometimes they target the cell wall. So whatever target was there of the antibiotic, it won't be targeted because the MDR1 protein is physically excluding, it is pumping out the antibiotic. And that is why MDR1 protein, if it is present on the bacterial cell provides antibiotic resistance. So this is how MDR1 protein, which is a glycoprotein, which is actually glycoprotein, it plays a role in multi-drug resistance. So what happens is that in certain tumor cells, if they get mutated and they start expressing this MDR1 protein, so now tumor cell also, they also get resistant to antibiotics, to get resistance to those particular type of drugs. And that this is how the chemotherapy fails in the treatment of cancer. When we do the chemotherapy, and multiple times the cancer is relapsed in the patient, and when this cancer relapses, certain times it undergoes certain mutations, and if it undergoes mutation and forms, it starts expressing this MDR1 protein, so it becomes difficult to treat the cancer, because now all of those tumor cells which are going to express this particular glycoprotein, they are going to develop the multi-drug resistance, and now the chemotherapy won't work there. So this is how the tumor cells, they geniusly bypass the ways, the killing treatments that we are going to do on them. So MDR1 protein, this, the thing that you have to remember is that it is essential for providing the antibiotic resistance. Whenever there is MDR1 in any type of cell, it is going to develop the multi-drug resistance. So if the protein starts expressing in bacteria, so now the bacteria is antibiotic resistant, and if it starts expressing in the tumor cells, so now the tumor cell has developed a multi-drug resistance. Why? Because MDR1 protein is going to pump out antibiotic from cell. Okay, so this is a very important example of this. The other example is TAP1 and TAP2. Well, we'll learn about TAP1 and TAP2 in an immunology lectures, but I'm just listing them down here. I'll recall this when we will discuss about immunology. Then the other example is a very unique protein known as CFTR, which is, cystic fibrosis transmembrane conductance regulator. So the CFTR protein, this is very unique because it functions as an ATP gated chloride channel. So, so it regulates epithelial salt and water transport. So whenever there is mutation that causes cystic fibrosis, so whenever this CFTR protein gets mutated, that results into cystic fibrosis, and that is why the name is cystic fibrosis transmembrane conductance regulator. Because if this protein is there, if this protein is functioning normally, cystic fibrosis won't occur. Basically, in cystic fibrosis, the thick mucus is created and it causes lung infections. So this is very unique because it is ATP gated chloride channel. Okay, so this is very important. So these are the different examples of the ABC transport. And with this, we have discussed about the primary active transport in detail. So as discussed that the primary active transport uses the ATP hydrolysis. So now by using ATP, the concentration gradient has been set across the membrane, and setting that concentration gradient, you require ATP hydrolysis. So after this ion concentration gradient is set, this ion concentration gradient could be used so that whatever energy is released, that energy will be used for performing the transport of other ion, and that is what we call as the secondary active transport. So unlike the primary active transport where we are directly hydrolyzing ATP, so we are breaking the phosphate-phosphate bond, and whatever energy will be released, that energy will be put so that molecules move against their concentration gradient. But in the secondary active transport, what do we do? That, let's say, let me explain it in a detailed way. So let's say that this is a plasma membrane, and now there is let's say almost equal concentration of sodium ions. Okay, so these are sodium ions. So the concentrations are the equal. So whatever sodium, let's say there is X micromolar concentration on the outside of the cell and X micromolar concentration of sodium ions inside the cell. So whatever concentration gradients are, they are equal. But now we have pumps here. So now what those pumps will do, these are the ATPases. So they are going to hydrolyze ATP. So they are going to do the primary active transport. They will hydrolyze ATP, and that whatever energy will be released, that is going to move sodium ions in certain direction. Let's say that the sodium ions, they are moving outside the cell. So let's say that this is cytosol and this is extracellular matrix. And so let's say that this is sodium potassium ATPase pump, and this just if it does this just kicks out the sodium from the cell. So now the concentration of sodium will be higher on the outside of cell, right? And inside of the cell it will be lower. So let's say that now the concentration of sodium ions outside the cell is X plus Y. So obviously X plus Y is going to be greater than X. So the concentration gradient is set for the sodium ions will be from outside to the inside. So this is the direction of concentration gradient, right? So for the movement of sodium ions in opposite directions. So for the movement of sodium ions inside to outside, we will need energy. But let's say that we move the sodium ions downhill in the concentration gradient towards the concentration gradient, means from the extracellular matrix to the cytosol. So this is going to follow the laws of nature, the laws of physics. So this is going to release the energy, right? So this is going to be the exothermic reaction. So this will release energy. So whatever energy is released, let's say that we put that in a good use. So let's say that I want to drive the transport of certain other molecules, let's say chloride molecules, or let's say potassium ions. So if I want to move those ions from here to here or towards their uh, to against their concentration gradients, let's say that the chloride ions are more towards the no, chloride ions, they are more in the cytosolic part. Let's say that they are more in cytosolic part, and I want to move those chloride ions against their concentration gradient. So what cell does, that cell just finds a better way. So what does cell do? It moves the sodium ions down the concentration gradient in the towards the concentration gradient. So now doing this it will release energy because it is an exothermic reaction. So whatever energy will be released, it will use that energy to move the chloride ions against their concentration gradient. So let's say that the concentration was low inside the cell, so it will just move the chloride ions towards the extracellular matrix, right? So this type of active transport is known as secondary active transport, because in the secondary active transport we are going to use the already established ion gradients, okay, the already established ion gradients, and we are going to move the ions according to the concentration gradients, so that whatever energy is released in that process, that energy will be used to move other molecules, to move other ions, right? A very unique and very fantastic way of doing the active transport, right? So that this is what we refer as the active transport. So here you can see that in the active transport, two types of molecules are moving, and that is why it is also referred as co-transport. Okay, so depending on the direction of the movement of the both molecules, let's say that the both of the molecules, they are moving in single direction. That is called as symport, and let's say that one molecules move outside, one molecules moves inside. Means the direction is not it is not the same. It is exactly opposite. That type of co-transport is known as antipode, right? So let's just list down the examples of the secondary active transport. Okay. Secondary active transport, which is also referred as co-transport, because we are going to use electrochemical gradient which was previously created by the primary transporters, and by using those electrochemical gradients we are moving solids, the other solids, against their concentration gradient. So let's just take an example. SGLT1. Okay, this is a sodium glucose transporter. Okay, if I'm saying symporter, means the both of the molecules, they are going to move in same direction. The two molecules here are the sodium and the glucose, right? So the sodium and glucose, they are both going to move in the same direction. So as I said previously, in the secondary active transport, we are going to use the already the preset concentration gradient. So here you can obviously guess that we are going to use the concentration gradient which has been set, the sodium ion concentration gradient, so that we can move glucose in the same direction to in which the sodium is moving. Right, so those types of symporters, SGLT1, sodium glucose symporter, though they are present on the intestinal epithelial cells, so that whenever the digestion is done, the glucose can be absorbed against its concentration gradient. Right, the molecules move, they are moving in opposite direction. So one molecule moves in this direction and other molecule moves in this direction. That is what we refer as the antipode. Okay, so the examples of antiporters, there are multiple examples such as the proton lactate. So there's sodium ion and calcium ion exchanger. Okay, then there's one more, fluoride bicarbonate exchanger. So all of them are the proton chloride exchanger. So they are the antiporters, means they are going to drive the movement of the two molecules in opposite directions. So with this I conclude the lecture. We have discussed about the different types of ATP transport that are seen in the cell. So let's just summarize the whole lecture once. Okay, so this is the table that I showed you in last class as well. In last class, we discussed about the passive transport, and today we discussed about the active transport. So depending on the type, depending on whether the ATP is hydrolyzed or not, that is the active transport is categorized into primary active transport and the secondary active transport. The secondary active transport uses the ion gradients which are set already by the primary active transport, and they could be the symporters or antiporters. So whatever flow is there, it is coupled flow, and that is why they are also known as co-transporters. The example that we saw is SGLT1 and NCX. So they indirectly use the ATP. So why indirectly? Because the ATP was already used by the primary transport to set the concentration gradients, right? And ATP was hydrolyzed, the ion concentration gradients were set, and now whatever ion concentration gradient was set, that will be used so that the secondary transport is done. So we say that the ATP was used but indirectly. Okay, so if the question arises that what type of energy source is used for bringing out the secondary transport, you would say the ion gradients. So the ions, they are moved according to the gradient downhill, and that releases energy, and that energy which is released is used to couple with the other molecule flow against its concentration gradient. Okay, the primary transporters are p-type ATPases, v-type ATPases, and APC transporters. All of them, they are going to hydrolyze the ATP, and they are going to drive the movement of the molecules against their concentration gradient. So p-type ATPases, they could move any type of molecules depending upon which type of pump it is. So for example, sodium potassium ATPases, it does the 3 sodium efflux and 2 potassium influx, right? But V-type ATPases, they are also known as proton pumps because they are only going to pump hydrogen ions. They are only going to maintain the proton gradient. Okay, so this helps in lysosomal acidification, in the neurotransmitter uptake in synaptic vesicles, right? So this is how the V-type ATPases, they are important. The P-type ATPases, they are the ones where the phosphorylation intermediate exists. Okay, that is the characteristics of P-type. While V-type, the V stands for vacuolar ATPases because they are present on the membranes of the vacuole-like structures like lysosome, endosomes, or synaptic vesicles. Right. While on the contrary to those ATPases, ABC transporter is very different because it has a transmembrane domain, nucleotide binding domain, and it is usually seen while doing efflux. So efflux means pumping some certain molecule outside the cell. So it usually pumps the antibiotics outside of the cell, and that is done by MDR1 protein, so which helps the cell to develop the multi-drug resistance or the antibiotic resistance. And CFTR, it is again important because if mutated, it results into the cystic fibrosis. So with this, we are done. We have discussed about active transport in detail. I hope everything is clear to you all. I hope that what are the differences between primary and secondary transport is very clear. In next class, we'll discuss about cell biology. We'll discuss about other things, the transport of the molecules in detail. We will discuss about the nuclear transport, how the nucleoporins, they are going to help in doing the nuclear transport. So with this, I conclude the lecture. See you in the next lecture. Till then, see you. Bye bye.